We'll deal with our questions. First, we'll go to our whiteboard. So, the ribosome. Here, ribosomes are appearing like the letter number 8. That is, just I'll draw and show you. In this way, you are observing the ribosomes. The ribosomes are having 2 units. This is first and this one is second. The upper unit, first unit is smaller one and the lower unit is larger one. And it is 50S and 60S we are calling. So here, the first one we are calling it as 50S and 60S. The upper unit we are calling it as 40S and 40S. You may ask me, sir, what you are writing this 40S, 50S and 60S? These are the units which are found by the Sedgwick. So, because of Sedgwick, his initial S letter is used here. And we are calling it as Sedgwick unit. Now, the ribosomes, in case of prokaryotic and eukaryotic cell, prokaryotic and eukaryotic cell, if you are taking, they are differentiated. Prokaryotic cell, they are having 70S ribosome, and eukaryotic cell, they are having 80S ribosome. So, the prokaryotic cell 70S, whatever is that, in both the cases 2 units, 2 sets per unit you are getting. The upper one is 40S unit, 40 sets per unit, and lower one is 50S unit in case of prokaryotic cell. But on the other hand, if you are taking a eukaryotic or advanced cells, in case of that, 80S type of ribosome you are getting. The smaller unit will be 40S and larger unit will be 60S in case of eukaryotes. And in case of prokaryotes, the smaller will be 40S and larger will be 50S. So in this way, the Sedgwick units are changed. Now dear friends, just see, the ribosomes are the sites of protein synthesis. Whatever protein is synthesized in the cell or the body, it is totally depending on these ribosomes. So we are calling ribosomes as the protein factories. Okay, so sites of protein biosynthesis we are calling. So you are already knowing how protein synthesis is taking place. In case of the DNA and the RNA, all these things you people study. But here we are only dealing with the ribosomes. Ribosomes are synthesizing protein and they are sites of the protein synthesis, or they are the factories of the protein synthesis. So in this way, the ribosomes, they are usually found all over the cell, usually freely found in the cytoplasm. Sometimes they are grouped together. Sometimes they are attached with the RNAs. So usually they are attached with mRNA, that is messenger RNA, and they are helping in protein synthesis. So we'll move to the next question now. We'll go back to our slide. Now see here, the first slide. Now, Which of the following plants contains centrioles in their cells? Where the centrioles are found? Which following plants they are containing? Just see. Option A, angiosperms. B, moss and some ferns. C, red algae. Then D, all of these. Here, angiosperms, all flowering plants you are calling them as angiosperms. Moss and some fern, here bryophytes and pteridophytes you are considering. Red algae, these are the algae they are found in this group. All of the above. Here the thallophytes and here all of these they are calling. Here the right answer is bryophytes and ferns, that is pteridophytes, that is moss and some ferns is the correct answer. Option B is the right one. Next, centrioles and centrosomes are present in cells of. In which cells the centrioles and centrosomes are seen? Now see, bacteria, cyanobacteria, green plants, animals. So see here, the centrioles and centrosomes are usually found in case of animals. Okay. Now here bacteria, here including bacteria in monoran cyanobacteria. It is also monoran that is helpful in nitrogen fixation as it is having blue-green alga. Then green plants, you are knowing. Okay, the animals here, the right answer is option D, that is animals. In case of animals, centrioles and centrosomes are seen. Next one. Now see, the function of centriole is. What is the function of centriole? Formation of spindle fibers, formation of nucleolus, initiation of cell division, formation of cell division. So here during cell division, the centrioles they are forming spindle fibers. So option A, formation of spindle fiber is the correct one. You may ask what is spindle fiber? Okay, now we'll go back to our whiteboard, then I'll show you where the spindle fibers are seen. Spindle fiber. Now in case of mitosis and meiosis, okay, after prophase, in prophase what is happening? That is the beginning stage, there the cell becomes ready, stores food, everything, okay, and the chromosomes whatever are there, they are very conspicuous and so on. So here, during the late prophase, these are the centrioles. Here, they are forming a fiber structure, and these fibrous structures, we are calling them as spindle fibers. Spindle fibers. And the chromosomes which are present with the help of the centrioles, they will be attached here. In this way, the attachment of the chromosomes you are observing. And the centromere of the chromosome attaches to the spindle fiber. These spindle fibers, they are undergoing a shivering movement. So as a result, the chromosomes, they will move towards the pole. On both the poles, the chromosomes are moving, and later this phase you are calling it as anaphase, and in case of telophase, they will be gathered at the pole. And then the cytokinesis is taking place, the division of cytoplasm, and cell will divide into two. And in later telophase, two cells are found in case of mitosis. So, in this way, Such a type of spindle fibers, they are helping to carry chromosomes from one place to another place. So, we'll move back to our slide. The function of centriole is the formation of spindle fibers is the correct one. Next. Next one is the function of centrosome is here inhibition of cell division, initiates cell division to increase protein synthesis. None of these. Yeah, nothing is done by centrosome. So the right option D is the none of these is the correct one. We'll move to the next. Centrosome is found in nucleus, nucleolus, cytoplasm, chromosomes. Centrosome is usually found in case of the cytoplasm of the cell. So, option C is the right one. We'll move to next. The main structure of centriole is. What is the main structure of the centriole? Now, 9 plus 3 fibrils, 9 plus 2 fibrils, 9 triplets, 13 globular subunits. Yeah, the correct option is option C, 9 triplets. So, 9 triplets is the right one. We'll move to the next. Now see, a plant cell usually differs from an animal cell in the absence of plant cell and animal cell. What is absent there? Just see. A. Ribosomes. B. Centriole. C. Mitochondria. D. Endoplasmic reticulum. The right answer is B. Option B. That is centriole. Centriole, the plant cell usually differs from animal cell in the absence of centriole. We'll move to the next. Controlling center of cell is. Which is the controlling center of the cell? Now, nucleus, nucleolus, mitochondria, ribosome. Here, the main switch of the cell, in the beginning I told you, the cell when you are taking, the main part of the cell or main switch of the cell or brain of the cell is nothing but the nucleus. So, here option A, nucleus is the right answer. So, controlling center of the cell is nothing but the nucleus. We'll move to the next. Now see, the role of nucleus in regulating the morphology of plant was discovered by. The role of nucleus in regulating morphology of plant was discovered by. The option Jesse, maize, then garden pea, Neurospora, Acetobularia. So, here the right option is D. Acetobularia is the correct one. Next. Nucleolus is found in. Nucleolus, where you are finding? Just see. Protoplasm, nucleus, cytoplasm, none of these. You are knowing it is option B, that is nucleus. In case of nucleus, the small rounded body you are observing, what you are calling it as nucleolus. Next one. The function of nucleolus is the synthesis of. So, DNA, mRNA, rRNA, tRNA. So, nucleolus is always synthesizing ribosomal RNA. That is option C, rRNA is the correct one. That is nothing but ribosomal RNA. Next one. Nuclear material without nuclear membrane is observed in. Nuclear material without nuclear membrane is observed in. Where you are observing without nuclear membrane? Okay, just see. Bacterial and green algae, cyanobacteria and red algae, bacteria and cyanobacteria, mycoplasmas and green algae. So I told you, nuclear material without nuclear membrane is the characteristic of prokaryotic cell, that is found in case of bacteria. You called it as nucleoid, or you are called it as incipient nucleus, or you also called it as false nucleus, that is found in case of option C, bacteria and cyanobacteria. So next one, the nucleoplasm is continuous with the cytoplasm of cell through. Now options: centriole, endoplasmic reticulum, nuclear pore and Golgi apparatus. Here the right answer is option C, nuclear pores. So what else? The nucleoplasm is continuous with the cytoplasm of a cell through. The nucleus you are knowing. Nucleus is covered with a double membrane nuclear membrane. Nuclear membrane is in connection with the nucleoplasm. So here the nucleoplasm is connected with cytoplasm through the nuclear pores. They are the connections in between nucleus and cytoplasm. Next one. The term nucleolus was coined by, who has given the term nucleolus? See the options: R. Brown, that is Robert Brown, H. Hooks, Bauman and Hansen. Yeah, the right answer is option C, Bauman's. So next one, karyolymph is what is karyolymph? Next, nuclear sac, SPM membrane, nuclear pore, none of these. Here, karyolymph is nothing but option A, that is nuclear sac. Nuclear sac is also called as karyolymph. Next one. The nuclear spindle consists of, what nuclear spindle is consisting? Just see option: 1 type of fiber, 2 types of fibers, 3 types of fibers, 4 types of fibers. It is containing option C, 3 types of fibers. Next one. Watson has calculated the nuclear pores of the mammalian cells to be dash of the total surface. To be dash of the total surface. How much percentage it is? Just see: 5%, 50%, 25%, 10%. It is option B, 50%. 50% is the right one. Next. The mastermind of the cell is, the mastermind of the cell. So, already I told, the brain of the cell I said. Okay, main switch of the cell I said. Okay, you are knowing. Just see the options, then you will come to know. A, protoplasm. B, nucleolus. C, nucleus. D, plastid. Yes, you are right. It is option C, nucleus. We'll move to the next. Paraamorpha is associated with. Paraamorpha is associated with. See the options: nucleus, chloroplast, mitochondria, nucleolus. It is associated with option D, that is nucleolus. So next one. Nucleoli are rich in. Okay, in what the nucleoli are rich? DNA and RNA, DNA, RNA and proteins, DNA and RNA. Now the right answer is option B, DNA, RNA and proteins. So next one. Histone proteins found in nuclei of eukaryotes are. So A, acidic, B. Basic. C. Neutral. D. Amphoteric. So it is an option B. Amphoteric. Sorry. Here acidic, basic, neutral and amphoteric. It is basic one. So histone proteins are found in nuclei of eukaryotes are option B, basic. Next one. The structure of nuclear membrane facilitates here synapses of homologous chromosomes and meiosis. Nucleocytoplasmic exchange of materials, anaphasic separation of daughter chromosomes, organization of spindles. So the right answer is option B, nucleocytoplasmic exchange of materials. So option B is the right one. Next, the nucleus has one membrane with pores, two membranes with pores, two membranes with pores through which substances do not pass, two membranes with pores through which micromolecules may pass. So, option D, two membranes with pores through which micromolecules may pass is the right one. Next. Nucleus is absent in. Where the nucleus is absent? Sieve tubes, companion cells, chlorenchyma, all the above. Usually, in companion cells, nucleus are present. Say, for example, here, xylem and phloem, if you are taking, in case of phloem, okay, the sieve tubes and sieve plates are present. And here in case of sieve tubes, the nucleus will be totally absent. So as a result, option A, sieve tubes is the right one. Next. The true nucleus is absent in. True nucleus is absent. Prokaryote, eukaryote, already you are knowing. Just see here, option A, green algae, B, fungi, C, lichens, D, bacteria. Bacteria is a prokaryotic type of cell. So in case of prokaryotic type of cell, the true nucleus is absent. That is membrane bond nucleus is absent. So only circular DNA you are observing and you are calling it as a nucleus, but the nucleus is a false nucleus, so we are calling it as nucleoid or incipient nucleus. So option D, bacteria is the right answer. We'll move to the next. So dear friends, that's all about the today's class. In coming class, first I'll explain you about the nucleus and then we'll move to the next class. So dear friends, we'll stop it here. Thank you.